Okay, so once again, good afternoon, and welcome to the keynote talk by Krishna Shenoy from Stanford as a highlight of our neural engineering theme. As you know, that theme, I know program chair, conference chair are here, but we have sort of taken over. I mean, this is the hot stuff in the field, and we have had fantastic number of posters and presentations, and I think we are sort of more or less ending with this thematic presentation by Krishna. So I want to make an introduction, and I must admit, I have to wear my glasses because it's pretty dense, lots of stuff there, you know, lots of good stuff he has done. So Krishna is a kind of genuine Californian. He even has roots in UCSD, as it turns out. He spent a year here. And our wonderful lunch, we realized he and his wife met at UCSD. So maybe I should send my kids to UCSD. Okay, so that's pretty good. And then he has moved to UC Irvine and then did his PhD at MIT with Fonstad. And then subsequently, he did his postdoc and senior fellowship at Caltech with Richard Anderson. And then he has been at Stanford for several years, almost since 2001. Krishna is one of those multidisciplinary person. He admitted that he veered away from anything that would make money and get him a really good job like electrical engineering to monkey work. But that means that he shows his multidisciplinarity from electrical engineering to neurobiology to bioengineering to the Stanford's crazy BioX and just about everything that goes with it. So he's one of the major figures at Stanford and in the bioengineering discipline. Krishna is a recipient of numerous awards, starting with his Hertz Fellowship as a doctoral student to subsequently Burroughs Wellcome, Sloan, McKnight, and so on. But perhaps the pinnacle of it, I would consider, is the NIH Director's Pioneer Award that he received. And you know, I thought that would be the highlight. But you know, knowing Krishna, I thought I'm thinking he might be proud of this. So he has Stanford University's postdoc mentoring award. We had lunch today, and you know, I invited 3 of my students, and said, students, say a little bit about yourself, and it became a kind of like a thesis exam, you know, because he asked a whole lot of questions, had a lot of suggestions. So he's a great friend, mentor, collaborator on all kind of research that goes around in this field. We have known him, many of us, through a variety of programs, particularly major, spectacular DARPA initiatives and programs that he's been part of. And you know, he is doing wonderful, interesting thing. There is this Stanford Institute for Neuroinnovation and Translational Neuroscience, for example. But he is committed to both basic neuroscience, some of the technology that comes to bear in working with behaving monkeys. He is now working with patients in very interesting multidisciplinary groups. So I would consider that he is really, truly, in NIH language, a pioneer. He has pioneered the neurosciences. He has had Nature paper within the last several months. So I mean, it just goes on. So I would say I think it'll be a treat. His work transcends from electrical to optogenetics and monkeys to devices, so it should be a treat. So please join me in welcoming Krishna here for his keynote talk. Okay.
Let me first of all check that everyone can hear me. Okay. Thank you, Nitish, for the very kind introduction, and to all the organizers for the wonderful invitation to give this talk. It's a great pleasure to be here today and to share with you some of our recent thinking and experiments aimed at helping the field as a whole move towards high-performance, clinically viable brain-machine interfaces. And roughly half of our group's work is aimed at pure basic neuroscience, which we think has good importance to brain-machine interfaces and has a lot of connections, but just to stay focused in today's talk, I'll be emphasizing just the brain-machine interface part. So to dive right in, the problem that we're attempting to address, and many of you in the audience, of course, as well, is that millions of people are unable to move or to communicate due to a variety of neurological injuries and disease. And this picture of Christopher Reeve is a somber reminder to us all that the debilitating effects of neurological injury, in his case a spinal cord injury, can be quite profound. Of course, he cannot walk, he cannot move his arms, or he could not when he was with us. But perhaps less appreciated, he also could not speak unfettered. It was hard for him to communicate. So how might we help people like this? Now, of course, we all hold great hope for stem cells and other neuroregenerative type solutions. But if we pause for a moment and look at this picture, a thought comes to mind, which is that maybe the thoughts, the plans, the intentions, the commands to actually move the muscles may still be intact within the brain, and they just can't make it out. So might it be possible, simply put, to eavesdrop in on the neural activity, translate that neural activity in whatever code or gibberish it might be in, and then use it to guide a prosthetic arm or a computer cursor on a screen? That's the basic proposition of what's called brain-machine interfaces, or BMIs, to interpret these neural signals and guide a prostheses. So to take the next step in terms of the level of detail, what we're seeing here is a side view of a person, or an animal for that matter, wanting to reach out for a cup of coffee. I think it's probably a person. I haven't yet seen a monkey drink a cup of coffee, but could happen. When you see a cup of coffee, you first of all look at it, and the coordinates for where that cup is come in through the temporal and occipital lobes in the rear part of your brain. Those plans are then elaborated and formed in the parietal lobe, and finally through the frontal lobe are sent down the spinal cord, unless of course there's an injury, as shown by the red X. So the idea is to listen in on neural signals, and just to state up front here, EEGs, ECOG signals, and intercortical signals are three of the types of signals that many of us in the room are looking at. And I think it's extremely important to look at all of those to try to understand how they can all complement each other and fit in. For this talk, I'll be focusing on only intracortical, where we're trying to attempt to record from individual neurons or small clusters of neurons. So as neural signals come out, we then need to interpret those neural signals with a decode algorithm, meaning turning it into something that can produce computer signals, signals of our construct that could be used to guide a prosthetic arm to reach out for that cup, or if we're thinking of a communication interface, to move a computer cursor on a screen to type out messages. Okay? So this is the basic proposition of brain-machine interfaces that I'm guessing the vast majority of you in this audience already know about, but I just want to help us all be on the same page before we forge ahead. Okay. Now, where do we turn from here? What I thought might be useful is to take a quick look at some recent advances, including a clinical trial. Second is to focus in on some challenges that we're facing as a field. Now, this list could be infinitely long, of course, right? This is not to exclude any challenges, but performance, robustness, and generalization are three that we've identified as things that our group might be able to contribute to. And finally, future opportunities, which simply put, once you get an arm out to something, you'd sure like to feel it, not crush it. How are you going to get artificial sensory information back into the brain? Okay? So let's start off with some recent advances. And just to put, again, this basic BMI concept into a block diagram form, just to keep it as simple as possible, we bring in sensory information, our brain processes it, and sends signals out through the spinal cord to drive the arm. And if that's no longer possible, all we're talking about is a bypass. We could think of it as a smart bypass, where we take out the brain signals through a prosthesis and drive motor output. And what we could think of building is a system where we tap into one or more of different cortical areas. In our case, we focus primarily on premotor cortex and primary motor cortex, though the parietal lobe and other areas are very much of interest as well. And then the prosthesis could be used to guide the location of a computer cursor on a keyboard and type out messages. So about 6 years ago now, Lee Hochberg, John Donahue, and colleagues were the first to try putting in a bunch of sensors, and by that I mean a silicon electrode array shown here on the right that measures about 4 millimeters by 4 millimeters and has 100 roughly 1 millimeter long electrodes. they implanted this into the motor cortex of a paralyzed individual resulting from a spinal cord injury. And from each one of those tiny electrodes was able to measure voltage versus time. That's the signal that's shown there. And what you're seeing in those very sharp deflections are individual action potentials from individual neurons or from a few different neurons. And what you can see is that sometimes there's very few of those action potentials or spikes. Over farther in time, there's many, and that could be when the participant is planning or thinking about not moving the arm or moving the arm to the left versus moving the arm to the right. And because each one of the neurons is picked up by the different electrodes has different so-called tuning across that population, you're able to get a pretty accurate picture of how the participant wants to move, okay? So that set of signals comes out the wire bundle to the connector that you see on this participant's head. The technician can then plug in a small, although I put small in quotes, it can always be smaller of course, preamplifier, an amplifier system that delivers those signals to one or more computers, that implements those decoding or translation algorithms, and renders back to the participant what you'll see on the next slide. Okay, so what I'm going to show you here is a brief movie in that it could help, again, bring us up to the same page in terms of roughly what the current state of the art is in a human clinical trial. So what we're seeing is a camera shot over the left shoulder of the same participant. You can see the preamplifier and the cable. The signals come out through the decoder, and just by thought, quote unquote, alone, moving the computer cursor on the screen. So this is about a 1-minute movie, and I want to just let it play through, and then I'll make a few comments before moving ahead to the challenges that I was talking about.
Okay, so here's the Cyberkinetics desktop. What are you going to do first?
I'm going to open my email first.
Okay.
I'm going to open the first one.
You're going to open the first one.
And it says, congratulations, you are doing a great job.
Very good.
I'm going to open the second email.
Okay.
This one says, hi, we'll talk soon.
Great. Now can you exit back to the Cyberkinetics desktop?
I'm going to exit.
You're going to exit. Excellent. So what's next?
Next, I'd like to draw a circle.
You're going to draw a circle. Wow.
So I'm going over to paint. Let me try that again. I erased it.
All right, let's try it again.
Here we go.
Whoops. You avoided the eraser that time.
That's the best circle I can do. I'm going to exit.
Okay.
I'm going to draw a circle.
All right. Excellent. Now let's try going back to the desktop. Very good.
Okay, so hopefully that gives you a sense as to the way in which, by thinking about moving one's arm, you're instead able to move a computer cursor on a screen. And something along the lines of the glass being half full and half empty is how I think many people would see this level of performance. The glass is half full, most certainly, because this is possible, right? It is an example of what may come in terms of helping patients that are extremely disabled. The glass is also half empty, perhaps, because there sure seems to be a long way to go before you may want to have this type of system used on yourself or on a loved one. Is the level of performance high enough? So this sort of helps outline and give a picture to the types of challenges we might want to focus in on. Okay? So first of all, let's see if we can increase performance. And what I'm meaning by performance in this particular context, performance can mean a wide range of things, but might we be able to help increase the straightness of those computer cursor movements? Instead of meandering like you may have seen in the movie, can you have it move along straight lines or along the path that you wish it to? Can the speed approach more of the speed that you or I could do with a mouse instead of having it move sort of slowly. And finally, and I think this is a really major one, can it stop precisely? A hallmark of good control is to be able to move rapidly but then come to a stop. And that has not yet been really exhibited clearly. So how might we go about doing this? Well, just to take a step back, as is the case with much of biomedical research, we have to rely on an animal model. And the good news here is that the animal model of choice in this field, before you go to humans, is typically the Rhesus monkey. Now, mice and rats and other animal models are critical, but just before you go into humans, usually the Rhesus monkey is very important due to the homology between the brain of the Rhesus monkey and the human, as well as the ability to train animals to do rather complex cognitive type tasks. And there's been a lot of research done over the past roughly 10 years. On the left, what I'm showing you here are various papers dealing with discrete target selection where you care about just the endpoint. And on the right, I'm just showing you some example papers on, again, these are all monkey papers that are related to moving the computer cursor along a certain path. And I'll focus only on this continuous movement of a cursor moving forward. So, how might we increase performance? So let's think again about the same block diagram we saw just a few slides ago, where sensory information, vision, in the case of the participant you saw, is coming in. He can see where the cursor is, and he knows where the goal is. That's brought into the brain where some processing is done, and usually that would go out the spinal cord and cause you to move your arm or move the mouse. Now, if that's no longer possible, as is the basic point all along here, what you want to do is design a decode algorithm. And how is that typically done? Decode algorithms are typically designed in what's so-called open loop. What does that mean? Have an animal make natural arm movements, record the neural activity, record how the arm is moving, and try to relate the two, okay? Many groups, again in this room, we certainly have built decode algorithms that do that very thing. But what might be missing is that that doesn't take into account feedback, the fact that when the animal moves the cursor a little bit off course, he's able to correct it, okay? And the second thing that's not taken into account is that you're no longer driving the real arm, the real plant, with those dynamics. You're instead driving what's shown in the red box, which is a prosthetic cursor on a screen or prosthetic arm, okay? And the location of that is being brought back around in the feedback loop. So simply put, we think, and down at the bottom are shown many references, many groups who have been on this thread for the last 3 or 4 years at least, where decode algorithms really must be designed in so-called closed loop. In other words, have the animal using the system and then tweak and understand and test the algorithm so that you're taking the entire system into account, okay? So this is rather a simple, if you will, engineering idea, but rather recently introduced into the neuroscience literature and prosthetics literature. Okay, now, what does that really mean in our hands? And I won't put up equations, but let me go one more level into depth to show you what is going on under the hood before I show you the results of some recent algorithmic work. And for those of you that were at Vikash Gilja's talk, who's here, earlier this week, this will hopefully look rather familiar. Okay? So on the left, we're showing what's called Innovation 1, which is neural control-based algorithm training. Now, what does that mean? What that means is go ahead and put in place the decode algorithm as shown in green, as you normally would, which does a good job of relating neural activity to arm movements. Start there. What else can you do? There are some other things you can do, but it's a good starting point, okay? And what that can do is give you the cursor, the neurally controlled cursor path shown in red, which meanders a little bit, not unlike the movie you saw. But because the animals are so trained to move to the target, and only when they hit the target do they receive reward, it's reasonable to infer that their intention is to move straight to the target, okay? Let's call it an assumption, but if that's the case, what we can do is we can go back and reinterpret the neural activity that we saw and draw in the blue arrows, whereby the neural activity at every time point is actually reflecting the animal's desire to move directly to the target. Okay? This is done only in the training period, not at runtime. Just to be clear, you still end up with a good old Kalman filter, which is our implementation in the end. But during the training period, you're reinterpreting what the animal is intending to do. Okay? And you can also add some other bells and whistles, like when you hit the target, surely the animal wishes to stay at the target, so reinterpret that neural activity to correspond to low or no velocity. Okay? Now on the right is a perhaps somewhat subtler point, innovation 2, feedback-based neural control algorithms. And what's shown here is a graphical model of effectively the Kalman filter, where P is the position of the cursor, V is the velocity of the cursor, and Y is the cursor itself that you might render. And what this is simply saying is that when you're in this feedback loop, as shown by the big red loop, the animal can see where the cursor is. There's no reason to infer that or to propagate its variance as you might other variables in a Kalman filter. Because you can see it, you can reduce its uncertainty effectively. Okay? And in the parlance of graphical models, this is called a causal intervention. That's why that one bubble and a lack of an arrow are shown in green. Now, how does this work? What I'll show you is a monkey looking at a screen in front of him, neurally controlling the white cursor, and a green target will come up, and his job is to go over there, dwell at that cursor location for 500 milliseconds. There's a little beep, that's his reward, and then he has to come back to the center. Okay, so the paths taken by that neurally controlled white cursor are reasonably straight, are faster, and I could spend a lot of time quantifying that for you, I won't. But the third thing seems to be the real gain, which is that when it hits the target, it can stop. It has good controllability. That's what I'd like to focus on in the coming 2 slides, just a little bit. So considerably better controllability or stopping, or what we can call faster dial-in times, okay? Now, let's say that you start off at this lower left target and you move the cursor out to the target displayed. Many algorithms can get you there pretty quickly, but many of them have it fly on through and maybe orbit around the target. So if you actually go look at the time from when you first of all came close to that target until you're able to finally come to a stop on that target, let's call that the dwell time. And I'll bold that here on this diagram, okay? And again, the reason that we're assuming that a straight line out there is the best is that's what we've trained our animals to do. Some very nice work coming from Aaron Batista and Byron Yu's lab. The bottom reference there has recently shown that animals can be trained to follow non-straight paths and also have very good degrees of control. Now, once we have this picture in mind, we can quantify things as shown in this graph. So on the vertical axis is the distance to the target. So if I start off here, and my goal is over here, I'm some distance away, and as I move in time, I hope to shrink that distance, and that's why you see the descent. And if you look at the green curve, you see that it descends pretty quickly, but that there's a large dial-in time. That thickened line corresponds to the thickened line on the previous time. So you get there within about 0.6 or 0.7 seconds, but it takes you another second to sort of fritter around the target before you can stop. That would be like doing one-finger typing where you get over here to the P key pretty quickly, but you can't come to enough accuracy to go ahead and select it, okay? Now, for reference, the blue curve shows the native arm. Our monkeys can also make real point-to-point arm movements, and you see that it is slightly quicker because that slope is slightly steeper. But look how short the dial-in time is. That simply says that the arm is really good. When you get out to a target, you can stop right away. Okay? And what's shown in between, no surprise, is the algorithm we've been working on, which we call refit, Kalman filter. And it is quicker than the existing state-of-the-art velocity Kalman filter, as shown by the red being to the left and slightly steeper than the green. But again, the real point is that it has a much shorter dial-in time. Okay? You can see this dial-in time plotted in a bar graph, again showing that it's much smaller than the green and close to the blue, which is the real arm. And I'd be remiss running a monkey lab if I didn't show you the second monkey with the same trends, because we don't believe anything, the field doesn't believe anything until it's replicated. Okay? Now, all I've shown you is point-to-point arm movements in 8 different directions. Let's train on that, but go to a harder task now, okay? A more realistic task. So what's shown here is a 6x6 grid of potential targets shown in yellow. Neurally, we're going to control the white cursor, and it's going to go out to one of the targets that we've cued the animal to treat as the target because it's cued in green, okay? And after I show you the movie, we'll come back to a point, which is that maybe if we vary the number of targets, more or less, or maybe if we vary the length of time that you need to have the cursor dwell on the target before it's considered selected, like hitting the key, maybe there's some sweet spot in that whole trade-off space. There is. We'll come to that in a second. Okay, but here's an example actually of an 8x8, so a 64-key keyboard. This is how it works. Okay, that's an error. You happen to be hearing one neuron, which isn't particularly informative because it's the whole population that drives it, but that's what you were hearing. Okay, so it's able to go roughly to 1 target per second. And what we can do then, as I was foreshadowing before, is we can go in and measure at different numbers of targets, 9, 25, 49, and 64. So a 3x3 up to an 8x8 target field. And we reasoned that if there were many targets, like if there were many keys on a keyboard, it might be easier to make a mistake and therefore performance would go down. On the other side, if there was only 2 keys or only 5 keys, it's really easy to make that selection, but the information transmitted per selection is so low, you would also penalize your overall performance. That's what's shown on the horizontal, and the black points are what we measured. What's shown on the vertical? What's shown on the vertical is the time that the cursor has to come over and dwell on the target before it's selected. If you wanted to make a system where you were really sure that you were on the right target before selecting it, you'd turn that dwell time way up, maybe to 850 milliseconds or even a second. But by doing so, you slow the whole system down. Again, it's like single finger typing where you have to dwell right above the key for a whole second before selecting it. On the other hand, if it's a very short dwell time, you might be making erroneous errors. Even on the way out to a target, you might accidentally dwell too long on an intermediate target and trip it. Okay? So what you can see is that there is a sweet spot. The color code corresponds to bit rate, which I won't go into detail on, but that's our performance metric. And what we can then do is operate at that peak, which is roughly 25 targets, 450 millisecond dwell time, and then start looking at longevity. So what we can do is we can bias ourselves at that set of points and run for roughly an hour and a half. That's what's shown on the left axis and in the gray curve. And if we integrate that performance, what you see on the right axis, right vertical axis, is the total information transmitted in kilobits. So over an hour and a half, you're getting about 20 kilobits, okay? Now, if this were a different IEEE society, and I was telling you about 20 kilobits over an hour and a half, I'd be laughed out of the room, right? I mean, this is not 4G, this is not 3G, this is like 0.0001G standard or something, okay? But there's a reason that these numbers are coming in the way they are, and I'll hopefully try to make that more intuitive to you in the next slide. Again, in a second monkey, the point here is that you can also identify the sweet spot. The red zone is in a comparable place. We can again operate at that point. Now, how to unpack this. This is a 4.3 roughly bit per second movie, okay, that corresponds to typing at about 10 words per minute, okay. We have not lost our minds, as far as we know. Our monkeys do not know about letters. They do not know how to spell, right? This is purely to hopefully convey what you could build for a human at some point. But if you build a 6x6 keyboard, you can certainly put all 26 letters, you can put special characters, and then we can go ahead and cue the target and have the animal hit it to start spelling out a message, okay? And what you're seeing here is what the level of performance and controllability that I was showing you in the past several slides might be turned into in terms of a usable interface, okay? And Paul Njoikin, who's sitting here, is overly influenced by his first C coding days, so hello world has to be the first thing that's typed, okay? It goes on to say something about yet 1,000 Shakespeares could they spell like a monkey. Okay, now that's performance, okay? And as many of us in the room appreciate, performance means really nothing without robustness. If we can't achieve performance over at least weeks and months, but essentially we got to have it for years and decades, we're not going to help people. So what can we say about robustness? How well can this work within days and across days? Okay? Well, what we can do is we can go in and have the animal make real arm movements to calibrate up our algorithm. If this were a human, we could just ask them to think about doing that. That's how it's done in the clinical trials. And we do that for a little bit of time just on one day. Okay, then we can train our algorithm, and then on a Monday, this is just an example week, we can record for about an hour at above 3 bits per second. Okay? Tuesday, we don't have any real arm movements going on, we just retrain, run for that length of time. About an hour and a half on Wednesday, Thursday. You know, our monkeys like to sleep in, that's why you're not seeing morning readings here. Okay, Saturday and Sunday to give ourselves the full week, okay? Now what does this mean? What this means is that we can go in for a week. Since we did this originally a year and a half ago, we've been able to go now for months, okay? But to make the point, if you draw a line at 3 bits per second, and you look at the fact that you can do that for over 11 hours across a week, and again, why are the monkeys stopping doing this at the end of the day? It's not that anything technically broke down, it's that they lose motivation, right? We're liquid rewarding them. If I were to try to do this task, I would lose motivation probably within 5 minutes, not an hour and a half, okay? But still, they can do 3 bits per second over 11 hours, which gives you about 14.5 kilobytes in total, 1.3 kilobytes per hour, which is about 2 book pages per hour, okay? So the detailed numbers here are obviously not important. The take-home message is that we think it's now possible to build systems where a person that has no other way of communicating might be able to communicate roughly 2 pages of text from a regular text per hour. Is that good enough? I don't know, right? I don't know that anybody knows. It really has such specific parameters to an individual patient. What we're trying to do is come up with a wider range of options, okay? But it's certainly way better than what can be done currently. What about across years? Across a week, across a month, fine. What about across years? Well, what I'm showing you here are 6 days of plugging into that connector that I was showing you on one of these silicon arrays, these Utah arrays or Blackhawk arrays. And across days, we absolutely see that the telltale sign of an action potential, a depolarizing event followed by a hyperpolarizing event, goes up and down across days, okay? Maybe some of it has to do with the contact resistance and how you screw into it, could be. Could be that it has the array shifted slightly with respect to the cells. It's a whole bunch of things, okay? But the point is that those signals go up and down. Now, here's the question. Do you care? Okay? If I'm over here wearing my neuroscience cap where I have to certify individual neurons, okay, and I need to know that those individual neurons are doing something specific, then yes, I care, because I want to be able to track those individual neurons. I want to hold them across days. I want to do plasticity and learning experiments like, say, Camarena does and so forth. But if I come over here and I put them on a prosthetics cap, I'm just after information. Maybe I don't give a hoot about individual neurons. Maybe what I should do is go throw down a threshold crossing. Let's just call it negative 4.5 RMS. And yeah, I'm going to lump together a couple of neurons. EEG lumps together potentially millions of neurons. ECOG lumps together maybe thousands of neurons. Here we're talking about lumping together maybe 1 to 4 neurons. Okay? Now, if you lump them together, will you still have enough tuning? Will this small cluster of neurons respond differently enough when planning to the right versus planning to the left that you have enough S and R that you can do your decoder across a population? The answer is yes. Okay? Again, there's no such thing as a yes versus no. Why do I say yes? I say yes because everything I just showed you was done with these threshold crossings. All the videos I played from our monkeys were using this threshold crossing method. Okay? And here's the final point. We've been doing this for over 2.5 years now, including from one animal whose implant went in 4.5 years ago, right? So this is now using, of course, 4.5-year-old array technology, and one would hope that the arrays are getting better, and we're able to maintain this level of performance. And as one indication of that, what I'm showing you on the vertical here is, sorry about this, a slightly different type of information called Fitts law information that takes into account the distance of the target and the constraint on how close you have to be, but it's an information metric across years. And one of our monkeys, J, you can see in orange, and one of our monkeys, L, in blue, and these data now go way off to the right side out to 4.5 years for L. And if you regress lines through this, you see zero slope. What's the take-home message? If you just do threshold crossing instead of trying to spike sort, you get a surprising, it surprised me at least, amount of robustness. You retain a surprising amount of tuning, and you're able to drive prostheses at world-class levels of performance. Okay? So, what about generalization? Okay? If we really want to help patients across a wide range of conditions, then we need to understand if what we've been learning will generalize to more natural behaviors and contexts. What do I mean? I've been showing you this picture of Christopher Reeve, who's a tetraplegic, meaning he is, not ambulatory. He was sitting in a wheelchair, he was relatively stationary, we could put an array, and we could do the things that I was showing you. But on the right is a broader class of patient that we would like to help, right? So you can imagine that this young mother wishes to be able to walk around, move her head, move her eyes, think about different things, all the while operating that left prosthetic arm to tie her daughter's shoes, for example. Now, why am I bothering all of us with this? I thought motor cortex, right here, was supposed to only care about moving the arm. So why are you talking about head movements or eye movements or cognitive states or body movements, all this stuff? Well, it's just not true. Motor cortex is absolutely influenced by a good many, if not all, of that list of things, if not more. Okay? It's in the literature, it's well documented. So as a field, we've sort of, we're as guilty as anybody else, closing our eyes to the fact that the signal source itself, whether it's EEG, ECOG, or intracortical, if you're in the cortex, you're going to get all these other variables that are modulating your activity. If you don't take them into account and understand them, they're going to be unexplained noise or it's going to hurt your performance. Okay? Simple point. So what we have to do somehow is go make measurements and understand what's going on in cortex in animals for the picture on the right. How can we do that? Well, I think there need to be 3 enablers. First of all, and one that's maybe particularly interesting to this audience, of course, as we all sort of have maybe some electrical engineering roots and so forth, is let's go build autonomous head-mounted systems. Let's build systems that in terms of the input can listen in on spikes in LFP, ECOG, EEG, EMG, even accelerometers. Let's then do some processing on that, some low-power analog electronics, have a microcontroller that works with a transceiver to wirelessly deliver that signal. So an animal could be walking around, you could be getting the neural activity. And ultimately output, if you'd like to be closed loop with this, is maybe that could trigger electrical stimulation or optical stimulation or even drug delivery, okay? And where I put the green check marks here show the types of systems that have already been built to date by the community and by us as well. Now, if you just have an animal running around, you're getting a bunch of neural data, you're still sort of out of luck. Because all you have is neural data, but no knowledge of how the animal is moving. Okay? You'll learn a lot of important things, but the picture won't be well instrumented like it is in traditional experiments. So the second enabler is Markerless, motion tracking. And this really is all about computer vision. Now, what am I talking about? Well, let's imagine a picture where instead of having an animal sitting in a little chair making these little arm movements like I was showing you in the first half of the talk, now we can have animals moving around their home cage playing with different things, all the while we're neurally recording, right? That's the telemetry system you're seeing there. But you also have a set of high-definition video cameras that are telling you about how the animal is moving, okay? Now, why markerless? What are markers? Well, you can, of course, go put on little infrared beads just like Hollywood does, right? You know, you put an actor in a suit, put on little beads. You can have commercial systems made by a whole variety of companies, track those beads. That's easy. Don't do that with a monkey, right? You leave a monkey alone with markers, you'll come back to no markers, right? They'll have all been deliciously eaten, okay? So you need to do this markerless. The more scientific point, of course, is you'll never be able to put a marker on every little segment you ever care about, right? It doesn't scale up. It would be way too intensive. So due to a variety of things that we could call Hollywood, by that I really do mean Hollywood is driving this technology. It's not just our little community, okay? They want to get actors out of marker suits as well. Computer science and musculoskeletal models, we hope to be able to infer what's shown on the right here, which is how the animals are moving. And finally, without some new computational storage paradigm, you're going to drown in data just like the early days of bioinformatics, okay? Except one could argue this is far worse than bioinformatics. You don't have 3 billion base pair and the combinatorics there. You have thousands of channels of time series data. You have multiple cameras. You're trying to correlate things to understand how the animal is moving, how the neural activity relates, and so forth. But again, fortunately, the computer architecture world is blasting forward with understanding how to do things in parallel. These are the graphical processing units, FPGAs, building these heterogeneous systems. And places like Google are, you know, among the very best at this. So those are the three enablers I would point to. Here's a couple of things we've done in terms of the wireless recording. So we can neurosurgically implant an electrode array, and as shown on the left, that leaves an electrode close to individual neurons like we've been talking about, and you can record. That then comes out to a connector as shown on the lower right, okay? And then what we can do is we can put a little aluminum canister around it so it's hardened, okay? And it's also a nice little RF shield. And then we can put a stack of boards, just a connector board, an analog board, a digital board, and a little wireless antenna, okay? And through the years, for the past 6 or 7 years, collaborators Teresa Ming, Boris Merman, Reed Harrison, Florian Sulzbacher, a whole crew of us have been building a succession of these so-called Hermes systems, where Hermes B had a couple of channels and a MEMS accelerometer. Hermes C, that I'll be telling you more about in the next 2 slides, has 20 channels of threshold crossings. Hermes D is 32 channels, and our latest Hermes E is the full 96 channels. So here is an illustration of the types of things that were possible a couple of years ago. So what if you went up to an animal in a cage, and what's shown in the 3 pictures below the word video is we put a Skittle, okay, or a healthier alternative, okay? They do like Skittles. And the animal reaches out, grabs the Skittle, brings it back in, and eats it, okay? And then we go and we look at the video and we hand label it, not what we wish to do. And this is Justin Foster, who's in the audience, and his talk earlier in the week shows you some more modern methods for doing this. But we can label reaching out in magenta and drawing back in. I'm sorry, reaching out in cyan and reaching back in magenta. And then you can label that on the neural data, and you can see a big difference in just one channel. Okay? So it's just an existence proof that even with one electrode, you could tell the difference between reaching out and reaching back. Okay? So it's sort of reassuring. What we can then do is go look across our 20 channels of data, and with any number of classifiers, we could automatically predict when the animal was reaching out or reaching back in. And without going into detail here, for this particular example, we reduced the dimensionality of the data down to a couple of principal components, and then with a support vector machine, could go ahead and predict, as shown in the little black ticks, when the animal is reaching out and back, and the real behavioral data is shown in the blue and the magenta. Okay, so it doesn't do perfectly. There are some errors if we went in and studied these patterns, but it does reasonably well, 90, 95+%. Now, that's an animal in a cage reaching out, reaching in. Surely we can do something that's a little bit more complicated as we build towards this ultimate vision. Let's put an animal on a treadmill. It's good for their health. We should all be on a treadmill, okay? But it gives us a little bit more regularity. It's sort of an intermediate step on the way to help us do the computer vision algorithm problem. And it's also a very interesting neuroscience question as well. And for BMIs, we can start asking the question, can you tell where the arm is even when the other arm and the legs and the tail and the head are moving freely? So it's sort of the first example case of seeing if we can tell how the arm is moving when other things are unconstrained. Remember, that was part of the overall goal. So what's shown with the letters A, B, C, D, E are some sort of stills, if you will, of how the arm was reaching out and then moving back in A, swinging back forward in B, pulling back again in C as part of a walking motion. D and E are when you reach out and get a food reward and pull back in, so we're mixing walking and reaching. The color bands on the neural data correspond to those behavioral epochs, and what each row in the neural data corresponds to is a different neuron. So you could squint and say, yeah, there's some difference between what's shown in green and orange, but it's really hard because you're effectively trying to peer back in through Poisson noise. So the better thing for me to show you would be to show you a couple of trial averaged channels. What does that mean? Well, in the upper panel labeled channel 7, if we just repeat roughly 20 times a walking cycle like this, and in the green is the period where he's pulling his arm back, the orange is where he's pushing his arm forward, then all of a sudden you see there's many more spikes when the arm is going back than when the arm is going forward. Okay? And channel 23 has a similar pattern. And then what we can do is if we go look at all 32 channels, now this was done with our Hermes D system, so it's only 32 channels, is that we can go ahead and classify those 8 different types of movements fairly well. Again, a set of standard classifiers would work. We happen to use linear or regularized regression and then maximum likelihood to classify them. In this particular scatter plot, the different colors don't look all that different in some cases, but if I showed it to you in 3 dimensions and started rotating it or thinking in higher dimensions, these clusters are quite separable so that we could tell these different phases apart with 95+% accuracy. Okay? So, in the last couple minutes on this section, then I'll just conclude with some future-looking work, and this is the optogenetics that Nitish was talking about. What are the next steps in wireless neural recording? Where is this going? And markerless motion capture, right? So I don't go into this in any way to say what we're doing. It's more to hopefully help try to lay out a roadmap that many people in this audience may be very interested in, and I think reflects how many in the field are viewing what the future might look like. So it's a good way to try to focus research. So this is a slide courtesy of Arto Nurmiko, who's sitting in the audience, who's a friend and collaborator. And what this is a fully implantable 100-channel wireless broadband neural implant. Everything I was showing you previously was head-mounted on top of the head, not designed for power considerations, for ever being implanted. It was designed for long-range telecom and low bit rate. And low bit error rate, okay? But it now appears possible to go ahead and create, what you're seeing in the picture is the standard Utah or Cyberkinetics, or BlackRock electrode array, okay, next to a titanium canister that's approximately 1 centimeter tall and a few centimeters on each side. And what's shown in the exploding diagram is that there's a rechargeable battery, and the whole can is titanium and hermetically sealed, right? So this looks very familiar to anybody in the medical electronics industry, right? We believe hermetically sealed when we see welded titanium. And this is fully functioning broadband 100 channel in 2 monkeys over 11 months. So what I would have thought would have been very, very hard, if not impossible 5 years ago, I think is now possible, which is that the head-mounted systems and the implantable systems can all finally collide and come together, okay? And this is just an example of the system I showed you working and recording from many channels, good spikes on each channel, and you can do some classification as well. The last slide on this topic is again trying to return us to this vision of trying to understand what's happening when you are moving your arms in an unconstrained situation. And what I showed you is a monkey's arm going out and back, and I showed you walking on a treadmill. And this is in collaboration with Michael Black, who's now a Max Planck director, was at Brown previously, and his student Oren Freefield. And what's shown here is an artist's rendition of a monkey, including fur, that then has a mesh put on it, and then has internal lower number parameter skeletal models put in, so that with a very few number of parameters, all things considered, you can vary different poses. So what you're seeing here is an operator moving a little circle that controls different parameters, and what you're seeing is the entire limb flex, okay? So now imagine if you have 8 cameras that are taking pictures of what's shown in purple, the overall animal's exterior. You could infer probabilistically what's going on with the internal models in some lower dimensional parameter space, okay, which is presumably the only hope of reducing the parameter space sufficiently to do that estimation problem. Okay, so in just the remaining 3 or 4 minutes here, before taking questions, if that's what might be desired, what about writing information back in? So let me now show you a brief movie, again from the Hochberg-Donahue group. This is the BrainGate 2 trial, where it's the exact same setup, different participant, same single electrode array, same connectorization, and what you're seeing is her move a robotic arm, okay? So what's happening is she's moving the robotic arm in 2 dimensions, just out and over in the plane of that table, okay? And going to pick up a bottle that has some coffee in it, okay? And then she issues a grasp signal, which is where you see that green dot turn yellow, okay? She's now trying to bring the coffee cup closer to her. Okay, this is still under neural control because that green dot in the corner is green. And now it turned yellow because she issued a grasp command and the computer went ahead and tipped it. What I'm describing here is some balance between letting you have free movement, but without potentially injuring the participant by getting too close, okay? So this is just to give you a snapshot of what I think is to come. People not just moving computer cursors, but paralyzed participants moving robotic arms. This work, some wonderful work that's just emerging from Andy Schwartz's group in terms of a paralyzed person moving a robotic arm, work by Richard Anderson that's about to emerge and so forth. There's a whole set of excitement going on in terms of really high performance arms, okay? Now, what is it about picking up a coffee cup that makes it hard? Well, you can get there and you can clamp down on it, but if you squeeze too hard, you crush it, okay? And it's often the case that you can't use vision because a lot of times it's occluded. So this is what makes different types of signals like somatosensation and proprioception very important. So is it possible, here's the question, to restore naturalistic sensation? The diagram shown here you're very familiar with is just reading out neural signals and driving, in this case, this blue prosthetic arm. But note there's a little pink rim around the hand, which you could imagine being sensorization, maybe not unlike what John Rogers was describing with the flexible electronics. You could sensorize that and wrap it around, okay, in his keynote talk yesterday. And what you'd like to do is to take the sensor signals, measuring pressure, for example, on the hand, send it back in through some sort of encoder, and then go ahead and stimulate the somatosensory and proprioceptive regions of the brain, okay? Now, electrical stimulation can absolutely do some of this. Electrical stimulation is extremely powerful. The paper by O'Doherty and Nikolaos is a wonderful one and shows exactly how one might do that. And many others, Aaron Batista, Lee Miller, Nico Hatsopoulos, Flip Sabes, are going after this problem with electrical stimulation. But it has some limitations too. When you stimulate, it's also very hard to record at the same time. The electrical currents tend to spread in surprising ways, new neuroscience is revealing, okay? And it's not selective. You can't control which cell types you are stimulating. So just by activating a ball of neurons in a sort of a spherical shape in the brain doesn't mean that it will necessarily be interpreted in that way, okay? Now, optogenetics, which I think most of you know about by now, if not years ago, activates and suppresses neurons, but only specific neurons that have been transfected by genetic methods, okay? And it can do so with millisecond precision, and because you're optically stimulating, it means you don't interfere at all with your electrical recordings if you do things right, okay? So we've been investigating this type of toolkit. What we do is we can, on the left, what's shown is go ahead and inject in viral vectors into Cortex. What this means is that a hypodermic needle can put in a microliter in a given location, and then you can pull up to a different location, put in another microliter, and you can do this across a field. You have to wait several weeks for the cells to uptake and to express the new channel proteins on the surface. These channel proteins that are light sensitive are called opsins. And what's shown on the right then is what you can do is you can drive laser light down an optical fiber, activating cells in a region shown in green, but again, only those cells that you've targeted, okay? For example, excitatory pyramidal cells, but not inhibitory cells, okay? Or the reverse. Or cells that are known to project to another area by virtue of doing a triangulation process, okay? So you can optically stimulate, you can record at the same time. This is typically done with what's called a conventional optrode, which is merely an electrode and an optical fiber glued together, okay? And the final thing to point out is what's called in vivo fluorescent measurements, which is that when you're doing this in monkeys, now that it's been established in the past couple of years you can do this in monkeys, is that you don't want to go in too early or be fishing around in the cortex. You want to go into the right place at the right time to make your measurements, and to do so, what you want to do is deliver light and see if the opsins have been expressed. And that's possible because of a Nobel Prize that came out of UCSD by Roger Chin, of course. These are fluorescent proteins, okay? So what's shown here in the top panel A is a histological slide in motor cortex. In green is shown, it's actually yellow, but is shown the cells that were labeled, including the descending fibers, okay? And what's shown in the middle there is the fluorescence return profile that we got, showing that this type of instrument works. For those optics aficionados, there's the actual optic light path shown for the instrument on the right. And then along the bottom is simply showing that we can do this across multiple areas: sensory cortices, motor cortices, parietal lobe. We can do so with different viruses, AAV, adeno-associated virus, or lentivirus. We can do so with different types of promoters, human Thi1, human synapsin. And we can do so with different opsins, importantly. Channelrhodopsin, so that when you shine light, you activate the cell. Halorhodopsin, so that when you shine light, you suppress the cell. And step function opsin that I'll show you in a second. So the reason for showing you this is that up to a couple of years ago, until the work out of Boyden lab at MIT, and then Carl Dijsselroos and our group's work, it was entirely unclear if this would work in monkeys. And there's very good molecular reasons to be very concerned. Might these overexpress, might they underexpress, and so forth, okay? What's shown is just some examples. On the left, you're seeing channelrhodopsin. On each row, you see spontaneous action potential firing. When you turn the blue laser on, it goes way up. And if you do that trial after trial, what you're seeing in the histogram below is that you drive the cell way up to about 600 spikes per second. So you can just radically turn on these cells. On the right is shown halorhodopsin, where the spontaneous activation is quenched, literally no spikes in some neurons, and shutting them down. Step function opsins are interesting. They're bistable. What's shown in the top plot is a 10 millisecond blue pulse that dramatically increases the baseline firing rate of the cell, but doesn't dictate where every spike gets laid down. So it's like you're jacking up the baseline firing rate, but without driving the cell or having it follow you around on a leash. You can also turn this off at will with a yellow pulse, as shown in the top plot. Now the bottom plot is the same thing again, same cell actually, where the blue ticks are delivered every 5 seconds, and every time there's a blue tick, it continues to integrate up the firing activity. And the reason it's doing that is every blue tick opens up more of the channels, causing there to be more depolarizing current, okay? And it basically integrates. Now, think about this from a medical device context. If you wanted to have an implantable device with low power, and you wanted to deliver low light power so as to not damage cortex, this is a way that you could pretty much chronically, with a low duty cycle, tick optically and prop up cell activity. Maybe not unlike what you'd want for a deep brain stimulator. And the final example here is C1V1, which is a red-shifted opsin, okay? And it behaves similar to the previous ones that I showed you, so I won't go through it in detail. The key difference here is this is with CAM kinase 2 alpha. This is an excitatory cell selective promoter, okay? So this represents the first time that we're able to go in and activate just excitatory pyramidal cells, for example, not the inhibitory cells, something you just absolutely could not do with electrical stimulation. Okay, so if you want to be writing in a code to the brain that it can understand, it's better to speak the brain's language and activate certain cell types, and we think this is a potential path forward. Now, in the last 2 slides before I summarize, I think there's a huge opportunity here for what we could call new optoelectronic instrumentation, right? So electrophysiology, recording only, is wonderful. I've certainly enjoyed it for 15 years. I think that time is more or less over. I think we have to also be doing optical stimulation. Otherwise, we can't causally perturb the system, and we really tie our hands. So to do that, we need a whole new set of tools. And what's shown here, again, is work from Arto Nomiko's lab. And what you can see in panel A here is an optical fiber where it's been tapered down, then gold plated so that you can record from the gold, and you can deliver light through the fiber. And then it's inserted into a stainless steel guide tube to make it very strong. And then there's epoxy reflow to make it very smooth, as you can see in panel B, the scanning electron microgram of the system. And basically, this does far less tissue damage than just gluing a fiber and electrode together, which looks like a barbecue fork. Obvious point, but if you're in monkeys or humans, that's a huge point. You don't want to be driving something like that through Cortex. It can do light delivery, electrophysiology, and also support the fluorescence measurements in Vivo, like we were talking. And the final slide here is, of course, you'd want to scale this up to arrays. You'd want to replace some of the electrical recording electrodes with optical delivery, or maybe replace everyone with the ability to both optically write in and electrically record, right? So these are just some examples of things that many of you that are extremely, extremely clever can come up with that could really support this brand new type of optoelectronic neurophysiology and BMIs. Okay? So in summary, it appears possible to increase performance by virtue of increasing straightness, speed, and stopping ability. We can get above 3 bits per second for continuous control. Robustness also appears possible, at least for hours and weeks. And in fact, in our hands at least, it appears possible out through several years. And naturalistic or artificial sensation may also be possible. At least optogenetics is possible in Rhesus, so we can now study it, because we can have these animals report to us what they're seeing and perceiving through some additional experiments. New instrumentation is needed, and future work will have to assess those perceptual qualities. So in take-home, reading from and writing to the nervous system may really enable a wide range of treatments, far beyond just the motor disorders. So in the most important slide I ever show, obviously the people, and as it's a little hard to go through person by person that way, here I'll simply say that in terms of the students and collaborators and postdocs, I've tried to highlight all of their respective contributions through the references at the bottom of each slide, and also special thanks to our supporters, including several funding agencies, DARPA and NIH, that are in the audience. Thank you.
Thank you, Krishna, for this really fantastic, eloquent presentation, for its clarity and also how cutting edge it all is. And it sort of seems to have like a beautiful orchestra come together in your hands, from old to new and science to cutting-edge engineering. So it's now for audience. Please, we are open for questions. So please come on up to the microphone so that we can take questions and everyone can hear you. And so I look forward to any of you who want to step up and have any questions. But Krishna, if I can get the ball rolling until the audience comes up, could you sort of point out the barriers? Because you want to tell us what should we do next. So I wonder if you can give us a little bit of the dark side or some major barriers we are facing.
Sure. Well, dark side is more fun to answer, I guess, than barriers. I'm not sure there are any dark forces. I think one thing I wanted to highlight is that designing the algorithms in closed loop seem to matter a lot. Designing algorithms that can take neural activity and predict regular movement, when you then take it into even the animal lab and see how it works, is a little bit dubious. I would then project even further ahead and say that it's very early days in terms of knowing which algorithms that work in monkeys work very well in humans. I think that there are likely some very big surprises there, none of which we know about now, but hopefully what we learn in early human translational studies will then really help set the agenda back in monkey labs. And maybe the final thing to say in terms of the technology opportunities, things are very, very far from done. I would say that this is just the beginning in terms of thinking about autonomous systems for monitoring and reporting. This obviously has a lot to do with the whole movement in terms of at-home wireless health monitoring that every major company in the valley around where we live is deeply interested in. Qualcomm here is a real leader in this. So these types of implantable autonomous reporting systems, new optoelectronic type systems, I think it's really a heyday for what engineering and biomedical engineering can do to really accelerate the field.
Hi, Krishna. It's
hey, Shane.
Good talk. Great to see you here. For these types of devices to really cross the threshold to more widespread critical use, they have to hit a certain risk-benefit and cost-benefit ratio compared to some of the existing technologies that are out there. And some of the thinking that you and your colleagues have done and some of the preparations for the translational studies you're doing, where do you think that threshold is in terms of capabilities for these things to start to take on the popularity of, say, the freehand systems or some of the other more successful neuroprosthetic devices out there?
It's a great question, which is ultimately, we're doing a variety of things in the lab, and even if they can help small patient populations, I think that's a long way away from what our medical device colleagues, our FDA colleagues, people that think deeply as you do about what it would take to make a successful commercial product. And by that I don't mean huge profit. I'm simply meaning a sustainable industry to really help people in the long run. And I honestly don't know for the types of systems that we're talking about where that threshold is. My personal motivation, so why am I not delusional as continuing to work on this, is I think that you sort of have to jump off a cliff and see where it leads sometimes. My hope is that through the ongoing renaissance that's happening in neuroscience, and that's why sort of half the work we do is there, in combination with the things that are happening in technology, will just change things in ways we can't predict in the coming 5 or 10 years.
But just a quick follow-up, but even with some of the very impressive demonstrations you're showing with basic communication and such, do you think the technology is now ready for more widespread testing with human subjects in these groups, and that we're crossing over that threshold now?
Well, so that I do think. So the question was, seeing some of the types of performance or robustness coming out of mini labs, not just what I showed here, but mini labs, is the time right for wider translational efforts? I am absolutely a supporter of that, as I am of ECOG, EEG, and intercortical. I'd like to see them all tested and actually quantified and put to the test, just like any other product is, right? I think that we're going to need to see a good diversity of the types of things that go on in terms of the translational efforts, i.e. it shouldn't be just one monolithic multi-site trial that consumes the entire nation and does everything in the same way. But I also think there is space and importance in being able to pool together resources so that we can go from an idea to having it tested in 10 patients within a month. I'm not aware of drug trials that would be interested in 3 patients. I mean, you know, there's got to be a scale to have that take up as well. But again, I'm no expert in any of this. I defer completely to medical device and FDA colleagues on these points.
So if I understood your explanation correctly, in your experiment of robustness of performance across many days, you retrain the decoder every day, right?
Yes, so 2 answers. So in what we showed here, okay, in what we showed here, what we did was every day we would come in with the previous day's settings, okay? We would then use that decoder to neurally control the cursor movement. And it often worked pretty well. But we would say, well, we can take the first few hundred trials and retrain, you know, just pause the animal for a minute, retrain the decoder and go forward. Okay, that's answer number 1. Answer number 2, in things that I did not show you here, that you will see at other conferences coming up in a couple months, shall we say, okay, hold the decoder rock solid stable and run for a long time, months and months and months, okay? And this is really to get at the heart of the question, which is EEG, ECOG, or intercortical, are they really that different, or are they about the same, or how do they really go? So we were sort of inspired, I guess said differently in our minds, retraining for 5 minutes compared to the overall apparatus is probably not that big a deal, but it should be interesting to know if we can hold the decoder exactly fixed. Those experiments have been done and they actually look quite encouraging, but stay tuned.
Okay, so do you, I guess what I wanted to hear from you is how much do you think we can rely on the subject's ability to adapt to the decoder instead of we are adapting our decoder to the neural activity?
Yeah, that's a great question. In fact, Jose is sitting right here and we talk about this a lot. You know, there's sort of these 2 extremes, and I actually don't believe in either extreme, right? I guess we represent the extremes, and I don't believe in you, and you don't believe in me, so it works. I don't believe in myself. You know, you could take one neuron as, you know, probably one of our all-time heroes, Eb Fetz, did decades ago, and train that one neuron to modulate in different and unexpected ways. And then you can scale that up, as Jose Carmena has done recently, and that's beautiful, and it shows exactly that the brain can adapt through plasticity and adaptation and work quite well. On the other extreme, what we sort of been looking at is to try to do the best out of the box that you possibly can, okay? I think only by putting those 2 together, and that's what I was really meaning, by fusing the 2 together can you have the best of both. Because there's no doubt that ultimately, whatever performance there is here, you could do better. Maybe plasticity and adaptation enables that. Or maybe that performance will roll off, and if you have constant adaptive signal processing running in the background, you could stave off that decline for longer. So hybrid solutions that mix optimally, I think, must be the way to go. Most engineering systems are.
So, fascinating talk, extremely impressive what you can get the central nervous system to do. But one of the questions that strikes me is that when someone loses control of the periphery, they not only lose the motor aspect, but they lose feedback as well.
Absolutely.
And so one of the questions, I guess, is that in designing these devices, the focus, at least that I've seen, is focus on everything's going through the cortex in this case. Are there advantages to thinking about trying to replicate more sophisticated peripheral mechanisms to minimize the moment-to-moment control that the cortex has to do?
Yes, I think the answer is most definitely yes. I mean, cortex primarily relies on driving, you know, spinal cord dynamics to get on out to the EMG and muscle drive ultimately. If we can take those types of musculoskeletal dynamics into account in the decoders, some of which we may be doing, but just in a black box sense already, But if we can do that more explicitly, I think that would be very interesting. Lee Miller, Nico Hatsopoulos, and others are going after exactly that type of approach. The focus on Cortex is part just because we like the Cortex. Just to be honest and frank. I mean, that's what brings a lot of investigators to an area. It's the area that one studies anyway. The better reason is that ultimately the injury can be so high that you really can't access the periphery whatsoever, whether it be the cord or the peripheral nerves, and you have to rely on Cortex. And that may actually have a good relation to Shane's question, which is, to really get through commercialization, to impact broader patient populations, you probably need to have systems that work at multiple levels or are ready to go at multiple levels. Sensation and feedback are, I think, critical. Okay, so.
Once again, Krishna, thanks a lot. It's a fantastic talk. We wish you good health and fantastic opportunity to carry on this brilliant work you've done. And thank you, audience, for joining us. It looks like the next session begins, so we probably can, leave the stage and let other session move in. Thank you all for coming.